Hi. You're ready. There we go. 1 second. I'm just setting up. 1 second. I should have it somewhere easier. Found. Live. Go. Hi, Andreas. Um, good luck. Good luck. Good luck. Good luck. Right. Okay. So, welcome for those of you who are now joining me. Welcome, welcome, welcome. And unless you have specific requests that you would like to do, guys, I've got some questions that I'm going to go through. So if you want to request specific topic areas, please do request away. Otherwise, I will just start going through some questions that I've already looked out. So if we go for, and actually share a different screen with you. Want to start with this one. So if you are year 13, then this is a question paper. This one is actually a year 12 question paper, but it's a really good question paper because it is looking at lactose intolerance. This weekly. So I usually do it on a Monday and a Wednesday, but I'm not going to be able to do it on. Oh, 1 second. I'm not going to be able to do it this coming Wednesday because I have to get myself prepared for going up to Scotland for the funeral. But I normally do it on a Monday and a Wednesday at 8:30. So this is a session that I also do live stream it so that you have access to it on my YouTube channel as well. So you can go and watch that. So if you are just joining me on the screen, then please do. On TikTok, please come over to my YouTube channel and you will see that you have access to join me in the actual live Zoom. And if you are wanting to ask questions, please do ask away. I'll try and jump between the two. But the reason I wanted to look at this question is this is a AS paper. And you can see here, so this one is the depth in biology. It's a 2022 paper, but I really want to look at this question 3. And the reason I am looking, I've just lost it now. Question 2. Question 3. The reason I want to start with this one is because if you're year 13, then this is a question where you will be asked, potentially in year 12 as an AS paper, you'll be asked questions which are up to the year 12 content, but in year 13, they could take this question and make it into an A-level question. So if we think about this and the information they've given us, so they tell us that congenital lactose intolerance. So straight away, they've given us some information. Lactose intolerance is where a person is born without the enzyme lactase. Now, if you don't have the enzyme lactase, you cannot digest the lactose down in the milk. And therefore, if you can't digest the lactose in the milk, what's going to happen is you are going to therefore not be able to utilize the glucose and the galactose in there. So I'm going to just remind myself, they've already told me that it's lactose. So if I understand that I've got my lactose, and my lactose is a disaccharide, and I know that that lactose disaccharide is made up of glucose and galactose. then already I'm starting to get my brain engaged in what is it that they may ask me. So then I need to think about, okay, what are they telling me? So the use of enzymes in technology. So this is where, when we look at this, enzymes in technology, this is where they'll link it to module 6. So as I said, this is a AS paper. However, this is actually talking about immobilized enzymes. So they could take that paper onto, or this question onto an immobilized enzyme question and assess your knowledge on module 6. So do be aware when you're looking at these AS papers that they can use these questions and just make them more adhere to your side of the specification. So what have we got? We've got milk containing lactose is going in. They've told us that they have a column of calcium alginate beads and they contain those lactase enzymes. So straight away, they're telling us that they are going to immobilize these. They're sticking them into a position and they're using the alginate beads to bind them so that they're not able to move. So if we understand that, let's see what they've told us. Well, again, a lot of the time they give you lots of hints. So you've got the glucose and galactose down here, but no lactose. So what does that tell me? Well, that tells me if there's no lactose left, then it has to be hydrolyzed. So already I can see that that's been hydrolyzed down, and therefore I now will look at the question. But if we are looking at how they could take this into a year 13 content lesson, then what they may do is they may expect you, just bring up my other PowerPoint on this, they may expect you to actually talk about the immobilization and how could it be immobilized. So what they may do is they may ask you to look at this and to talk about the fact that if you have got your immobilization happening, you have got that potentially we are going to attach these enzymes to some form of a support system. So we can either enclose it within a partially permeable membrane. So we could get it in that partially permeable membrane. So you can quite clearly see here I have my enzymes. That's what the blue is indicating. There's my enzyme. And here we have a phospholipid bilayer. So that partially permeable membrane, which is enclosing those enzymes so they are being kept in a specific area. We could use covalent bonding. So we could covalently bond them to an inorganic ionic carrier. Now, you do not need to know the names of specific examples. What they'll want you to do if they ask a question on this is they'll be looking at you knowing that it can be covalently bonded to that inorganic carrier, or it could be ionically bonded to the inorganic carrier. So there's lots of different examples that they could use. And a lot of the times they're going to ask a question based on this. They'll give you that information and expect you to pull out from in the stem of the question, whether it was covalently bonded or ionically bonded. And then they tend to ask you to recall the fact that it could be in a partially permeable membrane. It could be to do with adsorption. So it could be the fact that there may be hydrogen bonds to that inorganic carrier, or it could be entrapped in a matrix gel. So if they're going to ask any questions about this, they will be giving you some information. And just to reiterate this, learners are not required to recall the examples, but will be expected to apply their knowledge and understanding of immobilized enzymes in the context of biotechnology. So that is specifically being put into the specification for the exams this year. And that therefore is a way of showing you that although they've named some examples like glucose to fructose using glucose isomerase, or lactase, as is this example, glucose and galactose, glucoamylase, a way to break down starch. So we've got these dextrins down to the glucose syrup, or the aminoacrylase production of these pure samples of L-amino acids. These are examples that are in your specification, but they've said you do not need to recall them because they are going to give you examples on that. So do make sure when you're going through these types of questions that you're looking at what you need to know, you must memorize. Because remember, there's a very small percentage which is memorization. The rest of it is about your ability to apply the knowledge to the given situation. So let's move on and actually look at this question then. So what we can see here is this is asking us to name the type of bond that is going to be broken down by the enzyme lactase and describe what happens when that bond is broken. So this is important that we understand, as I've already said, we're going to hydrolyze it. So therefore, we're going to split it apart. How are we going to split it apart? So if we're looking at the glucose and galactose, we should know that we have got the glycosidic bond that's going to be broken. And what we should also note is that glycosidic bond that's going to be broken, that is going to be broken through hydrolysis. So a hydrolysis reaction. Now remember, hydrolysis means water splitting with water. So if we're going to do a hydrolysis reaction, we're going to split it apart, we therefore need to add water. So by adding the water, what we actually are going to do is we're going to end up with an OH group joining one of the sugars and the hydrogen joining the other. So the key thing to note is that the oxygen that was being held together between the two carbons, the only way we can split that apart is if we can actually convert that down. Let me just move this a second. Into two hydroxyl groups. So by hydrolyzing, by adding in the water, what we're going to do is we are going to split this apart, and we now have two hydroxyl groups rather than the glycosidic bond being held together. Okay, so let's keep going with this one then. Excuse me. A common symptom of lactose intolerance in adults is the creation of extra fluid in the large intestine. So this is where, when they're assessing your knowledge on this front, what they're looking to make sure that you do is that you actually note. Thank you, Paul. You note the fact that we were talking about a disaccharide, and we are going to break that down into two of the sugars. So what are we actually doing? We are going to affect water potential, because whether we're talking about the disaccharide or whether we're talking about those two sugars that we've actually hydrolyzed, what we have to understand is both of these are going to affect water potential. So if we are not breaking the lactose down into the reducing sugars and then absorbing them, what will happen is that is going to actually lower the water potential. So the common symptom is that it is going to end up with extra fluid in the large intestine. Well, they're expecting you to note that if we're talking about more fluid, we're talking about osmosis occurring. And therefore, what we need to understand is the lactose, because it is soluble, will lower the water potential in the large intestine. And because it is going to lower the water potential in the large intestine, that is therefore going to result in that water, try and write a bit slower, moving into the large intestine by osmosis. Okay, so let's have a look at what they've told us now. So the researchers indicates that reducing the dietary intake of saturated, so here we go, they've given us some information. Saturated triglycerides and cholesterol can reduce the potential risk of developing cardiovascular disease. So straight away, I already know saturation, if it's a saturated triglyceride, I know that there are only single bonds. Let's make sure we know that's to there. And the fact that it's a triglyceride, I know that that means it's got a glycerol and fatty acids. So I can quite clearly see where my glycerol molecule is, and I can see where my fatty acids are. So you can see that quite clearly in the diagram. So I would always annotate. Oh, and then the question goes on to say, describe how the structure of a again, biology is all about your wording. Many, and we've now got, that one, unsaturated. Well, if it's unsaturated, we know it's going to have double bonds, and those double bonds are between the carbons. So when you look at this, then what you're going to note is that you have got many double bonds between those carbons. Now, the other thing to note is if you have been taught this, you may have been taught this or you may not, there are different types of unsaturated fatty acids. There are what's known as cis and trans unsaturated fatty acids. Now, they don't expect you to know this specifically for your exam, but sometimes they do try and throw it in there to confuse you. So the way to know whether it is a trans or whether it is a cis, I like to think of it in the fact that if the hydrogens, one above, one below, looks a bit like a T shape. So that tells me if it looks a bit like a T shape, that is my trans, and this one looks like a C, and that is my cis fatty acid. So if I turn that on its side, I would see that that looks like a C. And the you're absolutely right. It's all about that Greek style. Understanding Greek, it would be a wonderful advantage for a lot of students these days. But because we don't tend to use it as often, although we do in biology, we don't tend to talk about it in the Latin and the Greek, you're absolutely right, or classical studies. What we don't tend to do is teach students that very much these days. So therefore, you've got to find what works to help you to actually remember it. Okay, so what's the difference? We've got many double bonds between those carbons. It's only a one marker. The other thing you could have said is if I'd have put a double bond here, let's say, and I removed that and removed that because I've got a double bond now between those two carbons, I have fewer hydrogen atoms. So another way that you could say this, and then the reason I was talking about the trans and the cis is because what you could end up with is kinks. So you might end up with, as you're doing your chains, you end up, if you've got a double bond, you could end up with a kink in the chain. And so therefore, what do we get? We get whenever there is unsaturated, polyunsaturated fats, what we tend to find is they are liquid at room temperature because they have more kinks in them, basically. And that's why you get plant oils, unlike our animal fats. Okay, so let's move on. Okay, whenever you get a graph, and this is something they love to do in the paper, is they love to try and confuse students by giving you two different scales. They'll never ask you to draw two different scales, but what they could do is give you a graph with two different scales to see if you're taking your time observing what's in front of you. So what we are told is some big long name that I'm not even going to try and say out loud because I say things all back to front. And actually half the time it doesn't really matter. here we do. We've got this condition, and this condition is going to cause high blood pressure, high blood cholesterol levels even in the person. Now, studies were carried out in the USA over several decades. They've told us the time period in which they've done it, looking at the overall death rates from cardiovascular disease and the percentage of the population with this condition in different age groups. So we can quite clearly see we've got age over 75. Let me just double check which one that one is. And I would advise you, as I've said each time, when you go into the, however, you need to do it the right way. When you go into the exam, make sure when you're doing it that you actually do make it easier for you to identify what it is that the graph is showing you. So I would actually highlight each of them a different color. We're not done green. Now the green one, I'm actually going to bring over here because this is to identify the CVD death rate per 1,000 people. And this one here is showing us the overall death for per 1,000 people. And also what we have to note is the percentage of age group with the condition. So do take your time when looking at a graph to really figure out what do each of the axes show and how can I then use that to help me. So we haven't even seen the question yet. That's the thing that I would always say is take your time and what can you see? So if we look at it goes up in age group, so we can quite clearly see age 20 to 44 has the lowest percentage of age group with the condition. Then it starts with the over 70s, or 75, should I say, and then goes above. Whereas for the first period of time, and this is the other thing to note as well, do make sure you're looking at the years. What years are we talking about? So up until the point of just before 2006, age 75 plus actually had a lower percentage of age group with the condition compared to that of younger individuals. So whenever you are looking at these, do make sure that you are evaluating, because I have a feeling anytime you get a question like this, there will be an evaluation. So let's just see what they say. A student looking at this data made the following statement: a fall in death rate from cardiovascular disease is due to a reduction in the percentage of people with the condition. With reference, now they've told us 20 to 44. So 20 to 44. So they've specifically said in this age group, in figure 3.3, discuss whether the student's statement is correct. So is it correct that a fall in death rate, now remember, death rate is the green, a fall in death rate from cardiovascular disease is due to a reduction in the percentage of people with the condition. So there is definitely a fall in the rate of the death rates, and we can see that there is a decline from 2006 to 2012. There is a decline in the pink. So yes, we can agree with that. However, there's a decline from 1994 all the way along to 2012 in the death rate. And yet, if you look at the numbers in the pink, and we're only looking at pink, from the 1994 period up to 2006, there's actually a rise. So when we see something like this, discuss, because that's what they've said, discuss whether the students are correct. You should always be thinking about support not support. So we've got a 3 marker here. So it's always important to make sure that you are obviously giving as much as you can to the question. So yes, word, try that again. From 2006 to 2012, there is a decrease. So we can see 2006 to 2012, there's a decrease in the percentage, and therefore also with a decrease in deaths. Now, I'm obviously writing this in very abbreviated style. Obviously, you give more. Yeah, absolutely. Now, on to the no. No, we don't say that the students are correct because from 1994 to 2006, we can see that there's a increase from 1994 to 2006. There is an increase in percentage, but a decrease in deaths. So we are disagreeing with that. Now, the other thing to note, they always, when it comes to a graph question, they're always going to allow data to support. So that data shouldn't just be reading off the table. The data should be giving it as a processed calculation. What I mean by that is that you are actually doing something with the data. So you are actually looking at what did it start at, what did it end with. So if you go to, I'll use one of them. So here, that's about 13. And here is about 16. So it goes from 13 to 16%. For the 1994 to 2006. But what happens, because I'm not just going to give them that data, I need to look at this, but oh, hold on a second. I need to come over to this one because it's my green. And that's one of the things I want you to note. When they give you 2 different scales, one of the problems that most students actually do is they always stick to that first scale. Go to the second scale, and that's the number that you're using. So we went to 2006. So we would have to give these numbers, which would be about 260 to about maybe 190. And so you want to give them that information. So therefore we had, what did I say, 200 and, I can't remember. How about that? Oh no, I said 260. It wouldn't be 260. It's too low. Oh. Let's try that again. So if that's 250, that would be about 278, maybe. Now, they're not expecting you to be bang on with those numbers, but they want a bit of a estimate. 270 to, what did I say? 190. 190. So that's between 1994 and 2006. So if you're going to do 180, 190, yeah, kind of between that number there, always make sure you are giving some bits of information. Now, they will always allow a bit of uncertainty. So if you're going to do the data and you're looking at it and it's something to think about, what would be the uncertainty in the data? So we can see that this percentage goes up to 10, then 10, then 10, then 10. So therefore, what are they going to allow? They're probably going to allow me a plus or minus 2%. If this is going up, and we can quite clearly see this is going up by 50, they're going to give me a plus or minus probably about 10. So that's important. The other thing to note, use the title units when you're writing the numbers down. So it's per 100,000 people. So don't abbreviate too quickly, because if you abbreviate too quickly, what will happen is you will lose marking points because you haven't actually given them the information they want. The easy answer, I would say, that you can always put down, but there's no guarantee that you will get the marking point. But the easy answer would be correlation does not mean causation. Now, a lot of the time, that answer is too generic for you to get the marking point. So students think that they should just throw it down and they'll definitely get the marking point. But it depends. It depends on whether this question is a high level question or whether it's a low level question. If it's a low level question, they are not going to allow correlation does not imply causation. So think about what other things could have actually, yeah, exactly. Iris, you're absolutely right. It's always the backup answer, but don't rely on that to get you the marks because it's never going to guarantee you those marks. So think about anything else that could actually cause cardiovascular disease. There's so many other things like, did you have a Mackie D's for lunch, i.e., is your diet a good one? Are you doing enough exercise? What is it that you're actually doing that could reduce that cardiovascular disease? Okay, let's move on then to the next question. Oh, went a bit too far. So here we go. The cardiac cycle here. We are looking at. Yeah, you're absolutely right. It's important to actually never abbreviate when it comes to graphs. If you're writing a conclusion or if you are taking data from the table, I would state never abbreviate at all. Don't get yourself into a bad habit of abbreviating. Always write it full, exactly whatever's in the table, exactly whatever's in the graph. You copy it word for word when it comes to the title or the units, because that way you're guaranteeing you're not going to lose a marking point there. But anything else, if you were talking about, I don't know, tobacco mosaic virus, for example, you'd write tobacco mosaic virus first off, and then TMV would be absolutely fine after that. However, when it comes to graphs, you write what they've given you. Okay, so we're being shown the pressure in the left side of the heart and the aorta during one cardiac cycle. So again, this is important. Look at what they've given you in the information. So we've got time in seconds. We've got pressure in kilopascals. As I said last week when I did this, I did talk about the fact that whenever you are doing the graphs and you're doing your practice, do make sure that you are using these as guides. This is how they want you to draw graphs if they ask you to draw a graph. So title with the units, specifically evenly spread out axes. £320 for over 12 hours tuition is actually, I get that for some people that may seem like a rip-off, but what you're actually paying for is not only my time that I'm doing here, which I do for free, by the way, but also all the planning that's gone into, all the analysis that's gone into it. And if you actually look at it, at the amount of tuition you get, it's £25 an hour, which is what you would pay for a university student, not someone with 20 years experience. So I understand for some of you that may seem quite expensive, but actually it is the cheapest I can do it for you, with keeping it that it's only a small group. So I understand it's not for everyone, but if you are interested, then obviously do get in contact. Thank you, Paul, but that's actually someone making a comment on the TikTok. It's interesting how many people want just the free stuff, and I get it, but what people forget and don't realize is that I don't get paid for these sessions. I don't get paid for any of my YouTube channel. I don't get paid for TikTok. I don't earn money by doing this. I do this for free. The only way I'm able to actually make a living is through the courses that I offer and through my one-to-one tuitions that I do as well. So I do this because I want to help as many as I can. I do one-to-one tuition, so please do get in contact, but I'm afraid I don't do it for £30 an hour. I'm afraid because actually, again, you get a lot of my planning time, a lot of my resources, all these resources I've actually built myself. And so yeah, all the diagrams, everything like that is all ones that I've created that are unique and specific to me and to the OCRA course and specification. So it's an interesting one because obviously people want things for free, and I get that because things are tight, but I have two children. I have to keep my business going as well. So I try and do it as low cost as I can, but unfortunately I need to feed my children and I need to live as well. And also what you get is a high quality session, not just somebody pretending that they know. Oh, seriously, my children eat fruit like there's no tomorrow. Okay, let's come back to this question then. So what you see here is the changes in pressure in the left side of the heart and aorta during one cardiac cycle. The student has described the events as shown. So at this point here, in the muscle wall of the atrium are contracting. This is caused by a wave of electrical excitation that starts in the atrioventricular node. So what I would be doing at this point here is I'd be drawing my little box. I'd be putting a little red dot there, and then I'm going to put a blue one here, which is going to come down. It's going to split, and then it's going to go up through the conducting fibers up through the walls of the ventricle. So then what you need to do is you need to then label. So this one here is my AVN. And if I go to my red, this one here is my SAN. So this is my sinoatrial node. And my sinoatrial node is going to send the excitation across the atria, causing the atria to contract. So straight away, I know that that is wrong. It should actually say the SAN. So it's not the atrioventricular node, it's the sinoatrial node. And that's important that I know that. How do I know that? Well, you can quite clearly see. And again, I would advise you trying to actually, oh, I knew it was going to do that. To highlight, no, let's try that again. Let's get rid of that. Because it's this type of graph. Let me see if I can go backwards. There we go, that's better. So that one's at the top. So the fact that my left atrium is at the top at this first section here, because we're looking at it, that bit right there. So therefore, my atrial pressure is greater than my ventricle pressure. So that's an important thing for me to note. Then it goes on and says, at B, the muscles in the wall of the ventricle are contracting. So here we go at B. The atrioventricular valves are open and the pressure in the aorta falls. So hold on, let's just remind ourselves again. They've said the atrioventricular valves are open. So if I think about this then, again, I'll do my box. If I now split it up, I've got my atria at the top, my ventricle underneath. So if I've got my atria and my ventricle, my atrioventricular valves, oh, that didn't change color. Let's try that again. Are in between the atria and the ventricle. So it's saying that the ventricles are contracting. If the ventricles are contracting, that's not going to open that atrioventricular valve. That is going to close the atrioventricular valve. So let's just carry on reading what it says. So at B, the muscles in the wall of the ventricle are contracting, atrioventricular valve closes, and the pressure in the aorta falls. Hold on a second. The pressure in the aorta falls. Well, if we're looking at the aorta and we're looking at the pressure in the aorta, we can quite clearly see it was falling before B. After B, it's actually rising up. So therefore, the pressure in the aorta doesn't fall. What we have is that the pressure in the aorta is actually rising. The ventricular pressure rises above that of the aorta. Yes, that is true, because we can see that if I use this color and then I come up and I follow it up, I can see that my green line, I didn't quite stick to the line, actually goes above. So that is true. Now let's look at the last one then. So the last one they're telling us is about C. So we're over here. At C, the muscles in the walls of the ventricle are relaxing. The semilunar valves are opening. Well, what we're seeing is our ventricle pressure is dropping below the aortic pressure. Therefore, those valves are not going to open. Those valves are going to close. And then the pressure in the ventricle is dropped. Well, that is true. It keeps dropping past C. So let's just see what this question actually wants us to do. Oh, there we go. Identify 3 errors in the student's description and write the corrections for each one. So this is something that they do love to do, where they give you a graph of a set of results. And what they then want you to do is they then want you to apply your knowledge to improve the information that is there. So that's assessing what you already know. So you should recognize this graph. You should know what's happening at each of the different points, which of the valves are opening, which of the valves are closing. So you can then note whether or not it is giving you the right information. Okay, let's move on then. Done. Let's see if this is it. Let's do this question, because this is a question that makes students actually get stuck when it comes to that application skills. And a lot of you seem to be struggling with this application skill. So let's look at this question then. So what do we have? We have got figure 6.1 shows a larvae of the gumleaf skeletonizer moth. And it is found in Australia and New Zealand. So again, straight away, what I'm noticing is they're already giving me my genus and my species name. So I can see that quite clearly already. What we've also got is some information that it has an exoskeleton. The exoskeleton is the external skeleton supports, interesting, and protects the soft tissue and organs of the larvae. It is shed periodically to allow the larvae to grow. So it's something that's going to support and protect. Each time it sheds its exoskeleton, the exoskeleton's head remains attached to its body, and these old exoskeleton heads stack up on top of each other. So here what they're doing is they're giving you information. They're giving you some information and they're wanting you then to apply your knowledge to think about how this actually would work. So they're stacking on top of the larvae is given the name the mad caterpillar because of their unusual adaptation. So straight away they're looking at, do you understand adaptations? And therefore, are you able to apply it to situations? So again, if you are a year 13 student, although this is an AS paper, how could they take this question and push it a bit further? Well, it depends where they've actually gone in the direction of the paper, but it could be that they're looking at you talking about how natural selection has resulted in this, and this one being different to another one that was potentially isolated. So then looking at speciation. So it's important whenever you are going through these questions that you don't just go, oh, this is the question. I'll try it myself. I'll allow the mark scheme answers to build my resources and think that's it. That's all they'll ever ask you. It won't. It will be about ways in which it could go if they were to use this question again. Now, they're not going to use exact same picture. They're not going to use exact same example, but they'll use something similar that could be the same, going along the same lines. Okay, so with reference to figure 6.1, suggests a purpose for the adaptation of attaching and stacking the old exoskeleton heads. So this is an interesting one because it's the same if you think about it with the reflex of when babies are born. So babies, when they're born, have what's known as the startle reflex, where they will spread out their arms, their legs when they are startled. And the reason they do that is to make them appear as big as possible. And by looking bigger, is a way in which they could potentially deter a predator from feeding on them. So it's a way to actually deter predation. So it's an adaptation where they are larger. So deter the predator. By deterring predation. The other thing to think about as well is if you are a a predator and you come along and you are feeding, it's an interesting one, and you are feeding on, you actually do try and feed on this stack, this exoskeleton, it's not going to give you very much nutrition. So therefore, anytime you see this type of organism, it could result in there being a way in which you are not going to feed on it because you don't feel, not you don't feel, that's not the right word, but they're not getting any nutrition from it. There's no nutritional value from the exoskeleton unless they have the enzymes to digest it, in case they went down that route. But what they're doing there is it's a way of showing, well, if they're not getting anything from it, that may deter them from feeding on something else that looks like it. So it could be this way in which you assume that there's no nutritional value in something that looks very similar when actually there is. It's that whole thing of stopping predation any which way they can. And when I went to the south of France last year on a holiday, we were walking around the trees and the cicadas were everywhere making crazy noises. But what we kept finding was their exoskeletons attached to the trees, literally just attached to the trees. And we kept at first glance, assuming that it was them, but only when we got closer did we actually realize it was only the exoskeleton which was left, which was great to see and very fascinating. But anyway, I go off on a rant, I always do. Okay, so what's it asking for us this time? It's asking us to give them the genus. Well, I'd already identified that. We'd written it up there, Uraba. So that is very clear when you look at that one. The table shows some taxonomic description. Now, I love the KPC or FGS. Now, you guys, whether or not your teacher is, yeah, possible decoy, absolutely, I agree with you. That's, I mean, it may not be, but it would make sense. It believes that, oh, hold on a second, you're saying possible decoy as in it would attract the predator to feed on it rather than the real live organism, right? Yeah, actually, yeah, potentially. So it could work in both ways. But going back to this one, if you have a cool teacher, that cool teacher will show you a really dodgy video, which you can find on YouTube. I absolutely love it, and I've shown it in every single class I've had, called the classification wrap. It's not good. It's kind of old style. Um, bad, bad making of movies. And actually, some of my classes in the past have tried to recreate their own version of it, which is quite funny. But it's always stuck in my head with the song, which goes KPC OFGS, KPC OFGS, and then it sings. I'm not going to sing it. But yes, that's the way I would always do it. So KP Crisps offers free gifts sometimes. Not many people will know about KP Crisps, though. That's the thing. If you said like Sensations or KP, what, peanuts nowadays, you don't tend to get very many KP Crisps. But yeah, finding whatever mnemonic helps you to remember it is always good. During kayaking, Paul caught octopuses for grilling supper. There we go. That's a cool one. So it just, I mean, I would never remember that. I've read that right now. Give me like 2 seconds. If I couldn't read it again, would not be able to see it. What we got? Keep pans clean or family gets sick. That's a good one, and that links to communicable diseases. Oh. Okay, so now what we need to do is we now need to put them into order. So Animalius, we've got kingdom, phylum, class, order, family, genus, species. So that's the one that is going with King Philip could order fritty green soup. Whatever works, whatever works. Fritty green soup. Where does the 6O2s come from in respiration? Like, where's oxygen actually involved? Okay, so Elle, to answer your question, the oxygen is the last electron acceptor for oxidative phosphorylation. So it's important to note that for the electron transport chain, you need the oxygen to accept the electrons. Otherwise, the whole ATC doesn't work. So you can't, if the ATC isn't functioning, the electrons are not going through the electron transport chain, there's no pumping of hydrogens, ions, there's no proton gradient being created. That means that they're not going to move back through from the intermembrane space back into the mitochondrial matrix, and therefore chemiosmosis won't occur. Oh no! Chidima, you're absolutely kidding me. Oh dear. Don't worry. I mean, to be honest, there's so many people that get them all the wrong way around. So interestingly, Paul, with regards to if they were studying the AQA specification, then terminal electron donor would be what they're looking for, but actually they want them to specifically say the final electron acceptor for OCR. So you want to get into habit of saying it's the final electron acceptor for the electron transport chain. And that therefore means that they'll get those marking points. Yeah, they are very picky on what they're expecting you to actually give them. Oh, here we go. On to a bit of the classification. So interestingly, this classification, they've gone on to the pepper moth, but actually they could have done something with regards to some random experience, a new organism that you'd never heard of, and that would therefore be looking at module 6. So let's see what they've told us. So we've got the peppered moth, has two forms. We've got the pale form and the dark form. We can see both of them. Then lots of information, lots of information. So it's really important when you're going through this that you are looking at the information they've given you. So we've got predators, birds, robins, and the thatch. We've got pepper moths. Avoid these predators by flying at night and resting on tree trunks during the day. Many trees have light colored lichens. So I'm just going to switch colors. So we have light colored lichen growing on them. However, many lichen are unable to tolerate high levels of air pollution. Without lichen, the bark of trees appears darker. Okay, so what have we got? Well, we've got a bit of information here where they've told us that they like to rest on the trees, and they are looking at the fact that they are light colored. So if we have camouflage, I've got to think this one through. Camouflage. Going on where something is obviously going to be hidden from those predators. So predators are going to feed on them if they see them. So if they see, i.e. are able to detect the moths, they are therefore going to feed on them. Those numbers are going to decrease. So detection will equal decline in numbers. So I'm just kind of giving myself some hints as to what might they be looking at when they ask these questions. So we're then told that these lichen are unable to tolerate high levels of pollution. So if the lichen, if there's high levels of pollution, high pollution, there's going to be less light, more dark moths. The dark form of the peppered moth first appeared in Britain during the, look at that, Industrial Revolution. What happened in the Industrial Revolution? Pollution. So therefore, we've got an increase in pollution. That increase in pollution was going to kill the lichen because they can't tolerate a high level of pollution. So if there's no lichen, the trees are going to appear dark. Therefore, the number of the light moths will decline. The number of the dark form is going to increase. Various surveys have shown that the percentage of the dark form of peppered moth population is higher in parts of England where there is lots of, well, that's obvious, heavy industrial compared to more rural areas. And here we go. Yeah, so it's an interesting one. So Paul, I would have always talked about that as well with regards to soot on the bark of the trees would also be a way in which it would help if the soot or the solid particles are actually binding to the tree. That would help with the camouflage because it would coat the tree trunks. So it is important that when you're going through this, that you refer back to what the question is giving you. They've given lots of information. What they want you to now do is apply that and your own knowledge to answer this question. So make sure you do refer back to what the question has said and not just throw in bits of information that you know in case it's not relevant and it doesn't give you the marking points. So again, that's where some students are like, I wrote loads of things on there and I didn't get the marking points. And what they've done is what they've given is not inaccurate, but it hasn't actually asked the questions. And this comes back to the fact that OCR are very picky. If they've given you a question like this, they really want you to note what it is that is expected. So what is the process of natural selection that has resulted in the distribution of the two forms of the peppered moth? So it's important to note, anytime I see natural selection, straight away, I will always do my keywords. So if I've got natural selection, I straight away go mutation, variation, selective pressure, survival, reproduction, alleles. So they're my keywords that I will always put down anytime I see the words natural selection, or if they're ever asking me to talk about natural selection. And then I'm going to take whatever example they've given me. So in this case, they are talking to you about the pale form and dark form of the peppered moth. If I go up here and I look at this one and think about the exoskeleton, and the fact that the larvae, we've got this exoskeleton, we've got the fact that these heads are being remaining attached, these old exoskeletons are stacking up. This could be that it was a random mutation that resulted in the enzymes, I guess, which would normally digest and break off that exoskeleton, not actually being produced. That resulted in the head staying on, the exoskeleton staying on. Therefore, the selective pressure would be, oh, hold on 1 second. There we go. Therefore, you would have it that they weren't being predated on, so they were surviving because they were appearing more as a threat, whereas another one, which didn't have the proteins not working properly. I'm trying to go down the route of where they may go with this. This is me just making it up on the spot, just to be clear. But they could go down that route of saying, well, there was an enzyme that digested it, that took the exoskeleton off completely. There was a mutation that happened that resulted in the exoskeleton not being broken away completely. That therefore resulted in advantageous selective pressure in the fact that it was now no longer predated on. Because it wasn't predated on, they survive. Because they survive, they can reproduce. Because they can reproduce, they then are able to pass on those alleles. So it doesn't matter what example they give you, if you've got those keywords that you can use and you can then apply it to the situation, and it will be much more straightforward than the one I've just tried to make up on the spot. It will all be in front of you to just tease out to get those points. So in this case, what are we looking at? Well, there was genetic variation. Where does that happen? Where do mutations occur? Remember, DNA replication results in spontaneous mutations. Now, again, those of you who are year 13, you need to think about this in the fact that if you have got genetic variation, these random mutations, the different alleles, different versions of the same gene, what are we talking about here? We're talking about the fact that those mutations, so the DNA sequence changes, therefore, the amino acid sequence changes, and that therefore could result in, I know, right, Scario, amino acid sequence changes, therefore you're ending up with a different primary structure and a different tertiary structure. Ooh, let's try that again. I'm glad I don't have to sit the exam, because I'm not joking. My writing is atrocious. These examiners would never be able to read my writing. And also, I get a sore hand from writing because I have to do it the way I do it. And so therefore, what it ends up is that I would I get messier as I go. My hand gets tired. Different tertiary structure. Now, I'm not answering the question. For those of you who are joining me and don't know what I'm doing right now, you have access to these papers. You have access to these mark schemes. You can go and look it up. There's no point in me just reading out what the mark scheme says. That's not going to help you. What's going to help you is me showing you how to apply the knowledge. For those of you who are just joining me, I do it normally on a Monday and a Wednesday. I'm not doing it this Wednesday because I've got to travel up to Scotland for the funeral. So I won't be on this Wednesday, but it's normally a Monday and a Wednesday at 8:30 on my YouTube channel. Yes, oxaloacetate. So how I do it, whenever I'm trying to think of those big words, because I'm dyslexic, I switch all my letters around. You'll notice it when I'm also talking to you and saying things out loud. My students are so used to it, they know what I mean when I say it. But I do it phonetically to try and help myself. So oxaloacetate. So try and get into habit, if you struggle with your spelling, to break it and chunk it into ways to help you. So oxaloacetate, citrate, acetyl coenzyme A are the three you need to know about for respiration. You're very welcome. So going back to this, in natural selection, random mutation has occurred. We've got different colors. In the Industrial Revolution, we know that it was more pollution. More pollution means there's going to be less lichen. So therefore, it's going to be darker. So the pales are going to be selected against, whereas the dark form is going to be selected for. So remember, there's two sides of this selection. So when we talk about selective pressure, those that are selected for and those that are selected against. So we would see a decline in the alleles of the less advantageous phenotype. That was the word I was trying to think of. So when we talk about as well, thinking about this, the phenotype, for those of you who are year 13, your phenotype is the physical characteristics that would be seen. So therefore, the dark form, the melanistic, the melanin that's being produced that makes it dark compared to the one which has less, which is the pale form. Okay. Right, where do we get to? Oh, here we go. Gotta love this one. So here we have two different, let me just decrease this slightly. So we've got the placental mole and we've got the marsupial mole. So we're being told we've got two different families here. So this is looking, because you're year 12, this is an AS paper. This is your module 4. And this is classification. If this were to go into an AS paper type of question, this would be about speciation. So this would be where they were asking you to talk about, well, what caused them to actually change. So it's important that you note when you are doing those papers that they can use these same examples, but just expect you to go into a bit more depth, okay? So what are we told? Where are they found? So this is important. They're found in North America, Asia, and Europe, whereas this one's found in Australia. So they're nowhere near each other. So that's one of the things to think about. They're nowhere near each other. What have we then got? We've got that they live in burrows in soft soil, live in burrows in soft soil. So they have the same habitat, but they're found in different places. That's the information they've given us so far. What do they eat? Well, they eat grubs and worms, grubs and worms. So they have the same lifestyle and they have the same habitat, but they're found in different locations. So then it's asking about, well, explain how this information is going to support the theory of convergent evolution. So it's important to note, first of all, this type of question, if they've said how figure 6.3, you must give them information from figure 6.3. So if you don't give them information from figure 6.3, you are therefore not doing as they've asked. I am shocked how many students don't see that in a question. So that's the first thing I would say to you. And then what they're looking for is for you to understand, well, what does convergent evolution mean? Are these closely related? Do they have a common ancestor? So one of the things for you to know of whether it's divergent or convergent is whether they have evolved from a common ancestor. So because we're talking about convergent, we are saying that there is no common ancestor. They are not closely related. And it's important, this key bit that everybody misses out, no recent common ancestor. So they have not evolved from a recent common ancestor because they live in different parts of the world. So the fact they live in different parts of the world means they do not share a recent common ancestor. So because they don't live in the same place, they therefore have to have adapted to the different environments, but they have the same niches. They live in the same habitats and they eat the same stuff. So because they live in the same habitats and eat the same stuff, they've adapted to the same niche, and therefore the way in which their bodies have actually evolved is the way in which it's best adapted to survival in the environment for which they live. So it is important whenever you are doing this to think about the information that they're saying. In answer to your question, is paper 3 usually harder than paper 1 and 2? Can't guarantee that. It depends. They will normally have one of the papers slightly easier than the others, but it really depends. I'd say paper 2 is probably the one that most students struggle with, because paper 2 is the one where they are assessing you on module 6 and also module 4, and a lot of students seem to struggle with module 6 and module 4. But it's not guaranteed. That it could be that paper 2 is actually a nicer paper this year and paper 3 is the harder one. But last year I would have said that paper 3 was the easier of the 3 papers, and I would have said paper 1, yeah, probably paper 1 I would have said was harder out of the 3. No. What I'm saying is there's no guarantee. It all depends on which way they've kind of written those papers and those questions. That's what's important. There's no guarantee, and anyone who says they know, they don't. They can make assumptions, they can make guesses based on their own research. So I've analyzed all the papers for the last since 2017 all the way through. I've actually gone back to 2016 all the way through, but I still don't know. And I wouldn't say that my predictions are spot on. I don't know. I can make a guess, but it all depends. They have to make sure that they've put enough low, middle, and high questions in there to assess the different levels. So it just really depends which way around they write it. But that is the end of the question paper. So I am going to finish tonight's session there. Say thank you for joining me, for those of you who've joined me. I won't be on Wednesday, so I will be back on Monday next week when I get back from Bonnie, Scotland. It's been a while, but it will be nice to see some families again. And so thank you and have a good evening. Thank you.